Bismillahirrahmanirrahim. Assalamu alaikum. Students, we were discussing about the water-soluble vitamins in our previous lecture. Today, the next water-soluble vitamin is the ascorbic acid or vitamin C, and it is the non-B complex vitamin. The vitamin C is synthesized by most of the plant and the animal kingdom in the uronic acid pathway. The vitamin C is essential nutrients for the human, higher primates, guinea pigs, and the fruit-eating bats because they lack the enzyme L-gluconolactone oxidase, which involves in the conversion of gluconolactone to the ascorbic acid. Both the ascorbic acid and dehydroascorbic acid have the vitamin activity. The naturally occurring vitamin C is the L-ascorbic acid, that is the reduced form. It has strong reducing property, which depends on the liberation of the hydrogen atom from the enediol hydroxyl group. This is the L-ascorbic acid, that is the reduced form of the vitamin C, and it is the L-dehydroascorbic acid, which is the oxidized form of the vitamin C. Sources, the rich sources of the vitamin C are the citrus fruits. You know as very well the family of the citrus fruits. Our tomato and guava are the good sources of the vitamin C. Vegetables also contain the vitamin C, and for example green pepper, cabbage, spinach, flour. The milk is the poor source of the vitamin C. RDA approximately 60 mg per day is recommended for the adults, however 45 mg per day is sufficient to prevent the disease of the scurvy. Absorption, distribution and excretion. Vitamin C is absorbed from the small intestine by a carrier-mediated process at the luminal surface that requires a sodium gradient. The transport resembles the sodium-dependent transport of the sugar and the amino acids. After absorption, the vitamin circulates in the plasma, red cells, and the leukocytes. It is widely distributed throughout the body. However, some tissues contain the high concentration of the vitamin C as compared to the others. The highest concentration of the vitamin C is in the adrenal gland, pituitary, and the retina in that order. It exists in the body largely in the reduced form with reversibly equilibrium with a relatively small amount of the dehydroascorbic acid. Under normal dietary intake, that is about 75 to 100 milligram, 50 to 75% are converted to the inactive compounds, while 25 to 50% is excreted in the urine as such. However, the vitamin C, like the other water-soluble, most of the other water-soluble vitamin, it is not stored in the body to a significant extent. Ascorbic acid is excreted in the urine as such or its metabolites. Here is the reaction in which the ascorbic acid in the reduced form is converted into the dehydroascorbic acid. It is a reversible reaction, and dehydroascorbic acid is converted into the other metabolites, and these are the keto acid and the oxalic acid. However, either the vitamin C is excreted as such or either in its metabolite forms through the urine. Structure and the synthesis of the vitamin C. The structure of the ascorbic acid resembles the monosaccharide hexose, and it can exist as the L and D isomers, and only the L form possess the vitamin activity. L-ascorbic acid. Ascorbic acid is being oxidized to the dehydroascorbic acid via the air, hydrogen peroxide, perchloride, methylene blue, percyanide, and this reaction is radially reversible by the presence of the reducing agents. The acidic property of vitamin C is due to the enolic hydroxyl group. However, it is a strong reducing agent due to the tendency to donate the reducing equivalents. It is most important function of the vitamin C. And the oxidation of ascorbic acid is rapid in the presence of the copper. According to the synthesis of the vitamin C, some lower mammals can synthesize the vitamin C from glucose by uronic acid pathway, while monkey, man, and guinea pig cannot synthesize the vitamin C because the deficiency of the gluconolecton oxidase enzyme. This enzyme is necessary for the conversion of the ketogluconolecton to ascorbic acid. Here is the reaction in which the gluconolecton is converted into the ascorbic acid in the presence of the gluconolecton oxidase enzyme. So the vitamin C, because the human cannot synthesize it, entire human requirement must consequently be supplied by the diet. Role of ascorbic acid or function of biochemical importance of the vitamin C or the ascorbic acid. As ascorbic acid function reducing agent, and also it has a scavenger of free radicals like antioxidant function. Ascorbic acid is radially oxidized to its biological equivalent dehydroascorbic acid, which can be readily reduced to reform ascorbic acid. However, there is the mechanism of action of ascorbic acid relative to its many activities is explained by its ability to undergo such reversible oxidation and reduction reaction in a large proportion of the reactions. The prime function of the vitamin C is to maintain metal cofactors in the lower balanced state, for example, iron in the ferrous form and the copper in the cuprous form. However, ascorbic acid has a specific role in the copper containing hydroxylases and the alpha ketoglutarate linked iron containing hydroxylases. These are the reactions. Dopamine beta hydroxylase, it is a copper containing enzyme involved in the synthesis of the catecholamines. You know very well catecholamines, norepinephrine and epinephrine from the tyrosine in the adrenal medulla and the central nervous system. During the hydroxylation of the copper is oxidized to the cupric reduction back to the cuprous, especially requires ascorbate, which is oxidized to the monodehydroascorbate. Here is the reaction in which the tyrosine is converted into the noradrenaline. Here the reaction where the dopamine is converted with the help of the dopamine beta hydroxylase enzyme into the noradrenaline, and the substrate is the dopamine, oxygen and ascorbate, while its products are the noradrenaline, water and the dehydroascorbic acid. Next is the peptidoglycine hydroxylase enzyme. Actually, the number of the peptide hormones have a carboxyl terminal amide that is derived from a terminal glycine residue. This glycine is hydroxylated on the alpha carbon by a copper containing enzyme called the peptidoglycine hydroxylase, which again requires ascorbate for reduction of the copper cupric form. Next, the iron containing hydroxylases. The number of the iron containing ascorbate requiring the hydroxylases share the common mechanism in which the hydroxylation of substrate is linked to the oxidative decarboxylation of the alpha ketoglutarate. Many of these enzymes are involved in the modification of the precursor protein called the post-translational modification. Proline and lysine hydroxylase are required for the post-synthetic modification of the proclagen to plagen and choline hydroxylase is also the conversion of the proline to the hydroxyproline with the hydroxylase enzyme and lysine into the hydroxylysine with the hydroxylase enzyme. However, these involve the post-translational modification of the proteins. However, the proline hydroxylase is also required in the formation of the osteocalcin and the component C1q of the complement system. The aspartate beta hydroxylase is required for the post-synthetic modification of the precursor protein C, and also the vitamin K dependent protease that hydrolyze activated factor 5 in the blood clotting cascade, while the trimethylysine and butyrobutane hydroxylase are required for the synthesis of the carnitine. In these enzymes, ascorbate is required to reduce the iron prosthetic group after the accidental oxidation during the reaction. Here it have simple vitamin C has simple catalytic role. In addition, there are many reactions which are performed by the vitamin C. It has a number of the non-enzymic effects as a result of its action as a reducing agent and the oxygen radical quenching. The important function of the vitamin C is act as an antioxidant. The water-soluble vitamin is only water-soluble vitamin, the vitamin C, act also as an antioxidant. Ascorbic acid is not only one of the strongest naturally occurring reducing agents known, but it can also serve as antioxidant in the several non-enzymatic reactions. It decreases the oxidation of the DNA and arrest protein damage, reduce lipid peroxidation and oxidation of the low density lipoproteins, and decrease the production of the extra cellular oxidants from the neutrophils. Here is the conversion of the ascorbate to the dehydroascorbate. During this reaction, it converts the glutathione in its reduced form, which act as a signature of the free radicals. Because of these action, it provides several health benefits, especially in the prevention of the atherosclerosis and coronary heart diseases. It is importantly associated with its antioxidant properties in the prevention of the cancer. For this, it may suppress the formation of the potentially carcinogenic nitrosamine from the dietary nitrite and nitrate in the stomach, which may explain its protective effect in cancer. However, several epidemiological studies suggest that the vitamin C exerts a synergistic effect with other dietary antioxidants, vitamin E and carotenoids, and this may have significant role in the prevention of the cancer, cardiovascular diseases, and the cataract formation. Also, the quantitative contributions of these components to the overall effect are not well known. Here is the mechanism where the antioxidant mechanism of vitamin E, vitamin C, and glutathione peroxidase. Actually, the tocopherol radical is relatively unreactive and ultimately forms the non-radical compound. Commonly, the tocopheroxyl radical is reduced back to the tocopherol by the reaction with the vitamin C from the plasma. The resultant stable monohydroascorbate radical then undergo enzymic or non-enzymic reaction to yield the ascorbate and dehydroascorbate, neither of which is the radical. Another functions of the vitamin C. Vitamin C is useful in the reconversion of the methemoglobin to the hemoglobin. Ascorbic acid enhances the iron absorption by keeping it in the ferrous form. However, vitamin C has a sparing action for other vitamins. Vitamin C also participates in the bone mineral metabolism. During bile acid synthesis, liver mitochondria, the 7-alpha hydroxylase reaction requires the ascorbic acid. During steroidogenesis, ascorbic acid is thought to participate in the several oxidation reduction reactions. This may explain the highest tissue concentration of ascorbate in the adrenal cortex. Vitamin E and C are used to slow the progression of the age-related macular degenerations. Vitamin C, the water-soluble vitamin, also antioxidant, acts together with the membrane antioxidants vitamin A and E to limit the extent of the free radical-mediated oxidative reactions. They prevent lipid peroxidation and may have anti-mutagenic properties. Vitamin C deficiency causes scurvy. It is characterized by the soft and spongy gums, teeth become loose, and there is poor wound healing. Many of the deficiency symptoms can be explained by a deficiency in the hydroxylation of the collagen, resulting in the defective connective tissues. The newly synthesized collagen cannot form fiber properly, which accounts for the prominent connective tissues abnormalities of the scalp. These are the deficiency manifestation of the vitamin C. The deficiency causes the scurvy. However, studies show that there may be benefits from the high intake of the vitamin C. At intake above 100 mg per day, the body's capacity to metabolize vitamin C is saturated, and any further intake is excreted in the urine. However, in addition to its role, vitamin C enhances also the absorption of the iron, as you know very well, and this depends on the presence of the vitamin in the gut. Therefore, you can say increase in intake may be beneficial. According to the recent studies, there is a very little good evidence that high doses of vitamin C prevent the common cold, although they may reduce the duration and severity of the symptoms. It was all about the vitamin C, its function and deficiency manifestations. Students, let me tell you one thing. Kindly consider Lippincott and Harper both for the detail of the water-soluble vitamins. Please kindly consult with both of the books of your Harper and Lippincott for detailed information of the water-soluble vitamins, because both are important according to your paper point of view and especially for the MCQs.